There we go. Can you hear me?
Yes, we can.
Excellent.
Share your slides?
I am trying to.
Okay.
Okay. I do apologize. I had to run to grab someone else's laptop, but it's an Apple and I'm not used to this. So I should be able to share screen. Let's see.
So Dori's going to talk to us about a novel population of synchronously active pyramidal cells in hippocampal area CA1 when her Apple laptop plays ball.
Can you see anything?
No.
No. Well, that's disappointing.
I mean, I have your slides emailed to me, so I can let me download them, and worst case scenario, we can do it kind of in an old-fashioned next slide, please, kind of.
Yes. It just, yeah, I don't know. I'm clicking share and it doesn't do anything.
Shall I share them?
Yeah, if you could do that, that would be great. Thank you so much.
So let me... Go with that. And then
excellent.
You see it? Cool.
Yeah.
Great. So I don't see you now, so you'll have to tell me.
I will. Okay. So sorry, everyone. Thanks for bearing with me on this. I will hopefully now talk about my research that I did at the University of Sussex. I'm currently in UCSD in San Diego, and also just moved here, so I don't have Wi-Fi yet, so hence all the issues as well. But we're here. So what I want to talk about is this population of synchronously active pyramidal cells that we found in hippocampal CA1. Next slide, please. So what I was doing basically is I was working on another project, and we had these Taiwan GCAM6F mice. So with the Taiwan, meaning that they're fluorescence in the excitatory cells. And we were doing the surgery, a hippocampal surgery, to insert a cannula over CA1. And what this does is it basically gives us a window just over CA1 where we can do two-photon imaging on these pyramidal cells in hippocampus. Next slide, please. So we had our mice in this virtual reality. So you can see it sort of, this is a 3D rendering from the back. You can see the mouse on the ball, and it can just move as it pleases, or it can stand still if it wants to. And as it moves, this virtual reality will move around it. And you can see the sort of lens there as well, which we use for the two-photon imaging. So I was doing this experiment and sort of sitting near the microscope and looking at my cells. Next slide, please. And I noticed these sort of very synchronous, weird cells during the recording. So that should be a video. I hope you can play it. If you can't, it should be okay, but
no, it looks like
that's fine. So we saw these highly synchronous cells. So believe me, they are very, very cool. I'll post a link in the Discord later on. Interestingly, we only saw these during stationary periods, so when the mouse was not moving, and the activity seems to be periodic. And so for these two reasons, I'll call them stationary low frequency oscillatory cells, and the rest of this presentation are slow cells. So we don't have the video, but I do on the next slide have a little graphic to show you sort of their behavior. So what you can see at the top here is 6 cells, the 6 that the arrows were pointing in the last slide. And you can see that the delta F over F here. So the more green is higher activity, basically. So you can see that these 6 cells during this sort of short rest period, it's about 3 minutes, just over 3 minutes, that they are active basically all at the same time every time. And you can see at the bottom as well, when you look at the average, which is the black trace is the average activity over these 6 cells, you can see that there seems to be some sort of periodicity to it as well. So they fire at a relatively sort of regular interval. So next slide, please. So I wanted to try to, I put that link on the wrong slide. Anyway, I wanted to try to see what percent of cells this was at first. So we took only 8 data sets because we couldn't see them in all data sets. And across these 8 data sets, there are about 6% of cells, so very small population. But you need to take into account that this was 8 data sets out of tens of data sets, so it's probably in reality an even smaller populations of cells that we see here. So my first step was to try to characterize them. So next slide, please. And first I wanted to look at their activities. As I said, by eye, it looked like they increased their activity during rest. So quantifying this, we showed that the activity between slow and the control cells, which is basically everything that's not the slow cells, is very similar during locomotion in green, but the slow cells increase their activity during rest, whereas the control cells decrease their activity during rest. So they have this a very sort of particular pattern of activation, essentially. So this was the first question I wanted to look at, which was the stationary sort of a characteristic. The next thing was the periodicity. So if you could go to the next slide. So used a Welch's method FFT basically to look at the power frequency spectrum, the power spectrum. And you can see here the slow cells on the left and the control cells on the right are plotted on the same y-axis. So you can see that the slow cells have a much, much higher activity or much, much higher power in this low frequency. So the x-axis only ranges from 0 to 1. When I control for the number of cells, because the slow cells have a much lower number, which creates like more noise in the average, when I controlled for that with the control cells as well, this particular band of interest between 0.1 and 0.4 hertz emerged. So here we could see that basically there's this band of interest where these cells particularly fire. You can see a similar band also emerge when we look at the difference between rest and locomotion. Next slide, please. So a question, of course, that emerged because these are cells in CA1 is what are they doing in terms of place? Because CA1, famous for its place cells, wanted to look at the sort of overlap between these cells and the place cells. And found that there was to some extent, an overlap. So you get about 10% place cells, whereas in the control cells, a bit more, so about just over 20%, which is sort of what we would expect. Do note, though, that, as I said, it's a very low percentage of cells. So we're talking about 5% of total cells are these slow cells. Then 10% of them are place cells. So in a population of about 200 cells, this is 1 cell. So that makes it a bit hard to say anything conclusive about it. But there definitely seems to be some overlap. Next slide, please. So then what I wanted to do is see if they actually had a function in spatial encoding. So first, looking during locomotion, when obviously the place cells are firing, we see that the place cells in pink have a higher spatial information than the non-place cells in blue, which is expected. But interestingly, there's no difference between the slow and the control cells. So they have the same sort of pattern of spatial encoding. However, so next one, please. If we look during rest, we can see that this pattern actually changes a bit. So we can see that suddenly the slow cells, so again, the left side, so the non-place cells in particular, but also still the place cells, have a very high spatial information during rest, higher than the control cells. So what we think this might indicate is that the slow cells maintain some information about space when the mouse is resting. And this fits with some there's not a lot of literature on these kinds of cells out here. That's why I call it a novel population. But with similar sort of research has found that this to be the case. But obviously, this is not very conclusive evidence. So this is something that we're still wanting to work on. Next slide, please. So just wanted to give the overall takeaway. So we have this novel population of highly synchronous cells, which we call the slow cells, which have increased activity during rest compared to the control cells. And they have they're particularly active in this frequency band of 0.1 to 0.4 hertz. And they seem to play a role in location encoding, particularly during rest. Next slide, please. So that leads me to thank the whole lab, as well as Caswell, Barry, and Roman Bourbelou, who have helped loads on this project. And also I need to thank Jenny for all your patience with all my technical issues today. And thank you for doing the slideshow for me today. I'm happy to answer questions here or over Discord.
Cool. Thank you very much. And well done for kind of keeping your cool when everything is. Let me... No, let's not close the video. How do I stop screen sharing? Okay, we'll just have infinite screens for a moment. There are some questions. Now mirrored infinitely. So how exactly did you pull out these slow cells from the rest of the population? Was it a criterion on oscillation frequency or synchronization at rest?
That's a very good question. So this is something this has been basically my main sort of issue with this whole project is it's very hard to, it's so easy to see them and it's very hard to sort of pull them out. So what I ended up doing with this is using this, what you said, the synchronization at rest. So basically I sort of did a recurrent algorithm where I add in the next most synchronous cell recurrently until I see a decrease in sort of the overall synchrony of this population of cells, and that seems to work. So basically, there's a natural sort of dip when I do this, and that seems to work really well in pulling out exactly this population.
So another question, can you explain what is the frequency power measure that you use? Is it the coherence of these spikes with different bands of the LFP?
No, so the frequency was, because it's calcium imaging, I can just take like this straight up the calcium data and just perform the fast Fourier transform on that. So we did also look at LFP, and there doesn't seem to be actually a very strong sort of coherence between the LFP and these particular cells. So this is in a different data set. We had LFP and calcium at the same time. But we think this may be due to just the low number of cells. So it seems like there may not be enough cells of them to really influence the LFP. Or another reason could be that the LFP is localized. It seems to be quite localized to certain areas of CA1. So if we don't have the probe exactly in that area, it's hard to measure it.
So I was going to ask you, and you're sort of touching on it now. You said that you found these slow cells in 8 out of tens of animals. Was there anything like, could you pinpoint differences in the region you were targeting or the mouse's experience, or like anything that would allow you to figure out why those animals and not others?
So I haven't actually looked at the experience yet. I do need to do that, but it seems to be particularly in some animals and not in others. So I think out of 12 animals, it was only in 3. I don't have, in my original data, I don't have enough information about the exact location, basically because we weren't doing this project to look at these cells. But I have now looked at the Allen Institute data. So they have some great data out there with some Neuropixels as well. And using that, it does seem to be very localized to sort of more ventral CA1.
Interesting. Cool. Well, if there are no more questions, which I don't think there are, I will thank all 3 of you again for really lovely talks. You get the sad solo clapping. And remind you all that you have a little over an hour now in which you can either enjoy your virtual meetup or go to the Flash Talks or follow on discussions on Discord. So thank you for attending this session, and we'll see you back in a little over an hour.